More absorption will be discussed during this short lecture. We're going to be talking about water, salts and minerals absorption, and we'll talk about absorption of water first. So this graph shows the fluid balance of the GI tract, and the GI tract is very well adapted to retain as much water as possible. On an average day, you will ingest about 2 liters of water. Obviously, as you exercise or become more sedentary, this is going to change. Your access to water is going to alter that, but we'll say that on an average day you take in about 2 liters. But that's not the only fluid that goes through the GI tract. You have secretions that add a significant amount to this, and the GI tract then has to reabsorb it. So you'll ingest about 2 liters. Your saliva is about 1.5 liters of secretions a day. Gastric secretions are about 2 liters. The bile and the pancreatic juices together make up another 2 liters, and your intestine will secrete about 1.5 liters all in all. And so you secrete about 7 liters, you ingest about 2, and then your small intestine will basically absorb about 8.5 liters of that, with another 400 mils being absorbed by the colon. And so the GI tract is very, very good at retaining water from ingestion and from secretions, so that actually what you excrete in the feces at the end is a very tiny portion of that, and in this example it's about 100 mils a day. So water freely diffuses through the GI tract, and therefore the absorption of the water is passive. And so what that means is that it's going to follow the osmotic gradient. If you take a drink of water, that water is going to pass into the vasculature because it's hypoosmotic compared to the plasma, and so it's just going to kind of diffuse through. Whereas if you were to eat a salty meal without much water in it, water is going to be dragged from the vasculature into the lumen of the GI tract to try and make it isoosmotic with the plasma, because that's what the GI tract generally ends up doing. It will move fluid in a direction to make the luminal contents isoosmotic. But water can be absorbed even in the absence of an osmotic gradient, or even against an osmotic gradient, particularly in the case of the colon. And the way that water flows in this situation is described by the standing gradient osmosis hypothesis. Here is a figure that shows the standing gradient osmosis, and I'll talk you through the principles of it. This is a way of creating sort of local osmotic gradients that allows the flow of water into the GI tract when there may not be any gradients between the luminal contents and the vasculature. And what happens here is that you have these enterocytes next to each other, and they have these intercellular spaces. And via the sodium-potassium ATPase, sodium is pumped into this area, and in order to maintain electroneutrality, chloride then passively flows into this area. And so this increases the osmotic potential of this area, and water then flows into this area, into this lateral space by osmosis, and this allows a hydrostatic flow of water. It requires energy, but this is one way that the body can bring in water against the osmotic gradient. These cells have tight junctions similar to those in the stomach, except in the stomach they're much, much tighter, and so you don't really get much water absorption in the stomach. But in the small intestine, these tight junctions are less tight, they are leaky, and so this allows another opportunity for water to come in, another pathway for water to come in, And so you can get a transcellular transport, which is what I talked about in the earlier slide, but also you can have paracellular transport, which is through the tight junctions and into these intracellular spaces. And this is important in the colon for concentrating the feces and removing a lot of the water out of it. Onto absorption of electrolytes and minerals. So this table shows you how sodium, potassium, chloride and bicarbonate are absorbed, and the mechanisms by how it happens. I want to make a point about the sodium absorption. It says actively absorbed. It's not through a direct mechanism. You have the sodium, potassium, ATPase that functions on the basolateral side of the membrane, and this lowers the concentration of sodium in the cytosol, and this allows a gradient then for sodium to come in with sugars and etc. And this happens all the way down the GI tract. So it's active because the gradient is set by the sodium-potassium pump. There isn't an actual direct pump that drags it in through the apical side. Potassium is passively absorbed until it gets to the colon, where it's sort of secreted. Chloride is absorbed, and it's also brought in exchange for bicarbonate in certain parts of the ileum and the colon. And then bicarbonate is absorbed early in the small intestine and then sort of secreted out later on. So the summary of that then is the small intestine is a net absorber of water, sodium, chloride and potassium, but it secretes bicarbonate, whereas the colon is a net absorber of water, sodium and chloride, but secretes both potassium and bicarbonate. Another way that minerals come into the body is what's called solvent drag. If you take a large volume of water in and there's solutes dissolved in it, as the water is absorbed, the ions and that are sort of dissolved in it come in then via this same sort of paracellular route. You know, you eat a hypertonic meal, large volumes of water are absorbed from the lumen into the blood together with the solutes that come in. And one good example of that is a hypertonic sports drink. You drink the large volume of water, and it's, you know, hypertonic to the blood, so as it goes into the small intestine, the water is absorbed and it'll drag then the sodium, potassium, chloride that's dissolved in it, and that will also aid in absorbing the sugar that aids in replenishing your body after whatever activity you've been doing. So calcium absorbed actively in the duodenum, but it's also passively absorbed by all segments of the intestine, the highest absorption being in the duodenum and the jejunum. And this shows how it's absorbed. There's a transcellular pathway where calcium comes in through calcium channels, and then this binds to calbinding, which is an intestinal binding protein, and then this will diffuse to the other side of the cell, where calcium is pumped out in exchange for sodium, or is actively pumped out via this calcium ATPase. But calcium is also thrown into these vesicles and then is passed across the basolateral membrane via exocytosis. The control of calcium absorption is regulated by vitamin D and parathyroid hormone. This is specific for the active calcium absorption. So if the calcium in the blood drops, this is sensed, and then you get the release of parathyroid hormone that then acts on the kidney, and this changes 2,5-hydroxy vitamin D3 into 1,2,5-hydroxy vitamin D, and this stimulates the synthesis of calcium binding protein and calcium ATPase in the enterocytes, stimulating then the absorption of calcium. Obviously, you saw in the previous slide that calcium needs energy then to pass over the basolateral membrane, and the transporter that does this, the calcium ATPase, is upregulated in the presence of 1,2,5-hydroxy vitamin D3, and the change to the absorption is shown here in this graph. You have duodenal absorption of calcium measured and the time in minutes, and as you add vitamin D12 to this, it almost doubles the absorption of calcium. The passive absorption of calcium is through the paracellular pathway, so going through these leaky tight junctions, and it's not under the control of vitamin C. So this will just happen down the concentration gradient in the small intestine. Magnesium is important for many enzymes and ATP-dependent processes, as it's an important cofactor for these things. The active absorption occurs in the ileum, and the mechanism isn't really well understood. But you also get minor passive amounts of absorption also through the paracellular pathway. So iron is an important mineral in your body, obviously for hemoglobin, but it's involved in your electron transport chain. Disturbances of iron metabolism can lead to anemia. If you're not getting enough iron in your diet, you don't make enough red blood cells, and don't have a lot of energy. But you can also have iron overload, where you ingest far too much iron. That causes complications. This has been seen in Eskimo populations that eat a lot of polar bear liver in the winter. And then you can have congenital defects like idiopathic hemochromatosis, where you just absorb excessive amounts of iron due to defects of iron transporting molecules, and this has also pathological effects. So there are two forms of iron in your diet. You have the heme iron, so hemoglobin, myoglobin, these cytochrome oxidase complexes, they are all of iron bound to heme molecules, but you can also take non-heme iron in, and these are dealt with in different ways. So the heme iron is absorbed by enterocytes, and absorption occurs by a facilitated transport molecule called a heme transporter, or it's endocytosed, and once it gets absorbed into the enterocyte, it's then split from the heme via a heme oxygenase reaction, and this releases free iron that then is transported via a transport protein into the blood, where it binds to transferrin and then is carried around the body in that form. You have non-heme iron that exists in two forms. You have ferric, which has a valency of 3, and these are generally not soluble at pH 7, so they're not transported. And then you have ferrous iron salts, which are soluble and that are transported. And vitamin C is important in reducing Fe3 plus to Fe2 plus, and therefore vitamin C contributes to iron absorption. The non-heme iron absorption is brought in through divalent metal transporters that are located in the brush border of the plasma membrane of the enterocytes. And there's also a reductase on the brush border surface that can also reduce iron 3 plus to iron 2 plus. And this aids then with vitamin C in making sure you get enough iron into your body. And then once iron is transported into the enterocyte, it is then transported into the blood, where it can be transported via binding to transferrin. In storage of iron, it's non-heme iron is deposited in all tissues, but specifically in the liver and the reticuloendothelial system. Intracellularly, iron is bound irreversibly to apoferritin to form ferritin, and this is the main storage form of iron in the body. And iron absorption is closely regulated by iron storage in the enterocyte and by plasma iron concentrations. And so when these fall, the absorption molecules are then upregulated.